Welcome everyone to the Seeing Math in Nature series with the Desert Laboratory on Tumamoc Hill. I'm Roseann Hansen, the Art and Science Program Coordinator, and I'm joined today by Paul Mirocha, the Tumamoc Artist in Residence, has been for over a decade, and Brian Enquist of the University of Arizona Department of Ecology and Evolutionary Biology. That's correct, Brian? We'll be doing formal.
Yes, that's correct.
Yeah, I'm going to do a little bit more bios for both of you in just a moment as we get going. So everyone, if you have questions during the workshop, please use the chat. I will be monitoring that. And also keep yourselves muted and your videos off, so that helps our bandwidth. I'll be putting resources and things into the chat, so you'll want to watch for that. And how we're going to work today is Brian will start out with a presentation about fractals and his work. And then Paul will lead us through some visualizations, because that's what it's all about. That's why we have art and science. It's about the art of seeing, the art of understanding everything, right? Through the practice of art. So you'll get to draw some trees and follow along with Paul. So let me say a little bit. I know we have some folks here who are new to Tumamoc as well. And if you're not aware, the Desert Laboratory was founded in 1903. It's one of the oldest, if not the oldest, continuously active, pretty much, field stations in North America. And it also has a rich cultural history, thousands of years of habitation by early people. There are hundreds of pit houses on the hill, and agave cultivation was common here. It is a historic landmark, and we have 860 acres of ecological reserve, and the field of ecology was basically born on Tumamoc, as was the journal Ecology was born in the library. And it's given, Tumamoc gave birth to also plant physiology studies, and arid land studies were born here. So welcome. Be sure to check out our website to learn more. And so first up, I'm going to introduce Brian Enquist. Dr. Enquist is a very accomplished ecologist. I just love your lab, your lab website, which I'm going to put the links in shortly, is just fascinating. Everybody, be sure you go there and check out Brian's his website. He's interested in understanding the origin and predicting the future of biodiversity. He studies organismal form, function, physiology in tropical and temperate environments, and uses theory, physiological function, and computational approaches to predict larger scale ecological, evolutionary, and ecosystem patterns and processes, which is so fun. So I'm going to turn it over to Brian. You'll want to unmute yourself there, Brian. And Paul and I are going to step away and make you the main speaker now.
Okay, can you hear me?
Yes, we're good to go.
Okay, excellent. So here, I'll share my screen. Can everybody see my screen?
Yes, we're good.
Yes. Do you see my main slide and not the presenter slide?
That's correct.
Yes, excellent. All right, excellent. And if you'll notice, I'm also doing some closed captioning kind of on this too. All right. So what I would like to do is kind of get started. This is a challenging but really fun topic. As you'll see, it's challenging because obviously we're dealing with math, but I'm presuming because all of you are here, you're interested in math or may have a background in math. And so I'm going to try my best to make this math light as much as possible, but as you'll see, I may slip a little bit. So, so if I do, please bear with me. All right, so for today, I actually want to do three things. I want to give you an overview of how to see the geometry of nature. And in the process of doing so, this is actually going to be a little bit of a history of this topic. And what's great about this Tumamoc series is this intersection between art and science. And this topic actually is really unique because I think I can kind of point to several examples where actually it was the artists kind of leading the way and pointing the way then to the scientists who then came in later and mathematicians to then kind of fill in our deeper understanding, okay? And in the process of doing so, I'm gonna hopefully kind of teach you another way to see the geometry of nature. Okay, and so I'm going to do an introduction to terms like self-similarity and fractals. Okay, and so we're going to be talking about self-similarity and fractals as it relates to the rules of life. And what I hope to kind of inspire in you is that the diversity of life around you is enormously complicated, right? And so that's kind of a fact of life, but yet kind of underlying that complexity, we can actually distill and see this unique geometry of life that appears to be governed by pretty simple rules, okay? So let's kind of get started here. All right, so I actually want to start kind of our story today with Leonardo da Vinci. And so many of you know Leonardo already for various different reasons. He really was this Italian polymath, right, back in the Renaissance. And he was he's a polymath because he was an expertise in many things. Not only was he an artist, but he was arguably kind of one of our foundational scientists, as well as making important insights into math, engineering, and kind of so on. And if you get a chance, I would highly recommend obtaining a copy of Leonardo da Vinci's notebooks. Not the original notebooks here, but actually the published form where they've gone through and kind of scanned all the different images and all of the drawings. And Leonardo was a very interesting guy. I think he was a little paranoid about people stealing his ideas. So he actually wrote, these are his notebooks. And if you notice, the writing looks kind of weird. He actually wrote mirrored as if he was writing in a mirror, and also backwards. The guy's brain must have been wired very differently. And so he wrote this way, apparently so that people wouldn't steal his ideas if they quickly looked at his notebook. All right, kind of an interesting kind of tidbit. But if you kind of scroll through his notebooks, they're filled with not only sketches of what would be some of his most famous kind of contributions in the art world, right? But also these amazing engineering ideas and how to build these really incredible, complicated machines, right? And so he was hired by various different kings and governments in order to kind of not only design weaponry, but also design new technology, right? But he was also an amazing natural historian. And if you scroll through Leonardo's notebooks, there are these amazing drawings of animal and plant anatomy and trying to figure out how to capture the beauty of not only nature, but also distill down, and this is kind of his engineering brain, kind of what the rules then of life are. And we're actually discovering more and more about biological insights from Leonardo by kind of scrolling through his notebooks and looking at his drawings and kind of fine detail. And one thing that Leonardo noticed is something incredibly important about nature that has really withstood then the test of time. And I think it's a testament to his art because he used these rules of what he saw in nature in terms of how light then would hit an object and how shadow would then be cast, as well as differences in perspective, but also the geometry of form and function. And in terms of life, and in particular within plants, he noticed a simple set of rules, but in particular, one rule that has become famous, something known as Leonardo's rule. And so Da Vinci kind of noticed that if you wanted to draw a tree, right? All right, so we all love trees when we do nature drawing, right? But if you want to draw a tree accurately, one rule that you could follow that would give you the perfect tree is something known as an area preserving branching rule. And Leonardo's rule says something like this, and you can see this here in his drawing. So if you look in the upper left-hand side, if you start with the parent branch, like the trunk of a tree, and you follow out all the secondary, tertiary branches as you go out to the tips where the leaves are, notice he's looking at these different branching kind of generations as you go out all the way to the tip. And what Leonardo actually noticed was that the cross-sectional area, as you go from this, what we call the mother branch to the daughter branches, is preserved. That is, if you take the cross-sectional area of daughter branch 1 and cross-sectional area of daughter branch 2, it actually equals the cross-sectional area of the mother branch. And this led to this amazing revelation that if you counted and summed up the cross-sectional areas of all the tips of a tree, it would equal the cross-sectional area of the trunk. Right? And that rule is known as area preserving. Okay, so this branching network we say is area preserving because the cross-sectional areas are preserved across the branching generations. Okay, and so we have a history of this in Leonardo's notebook. And if we actually study the trees that he drew in his various paintings and things, you'll actually see that they follow this area preserving rule. Okay, so in our research here at the University of Arizona, we actually study plants, we study trees, but we're interested in these rules, in particular the Leonardo rule, because they actually help us do a set of amazing things that we couldn't if we didn't have that rule. So that Leonardo rule of area preserving branches actually enables us to predict how much carbon is in a tree. It tells us how much carbon dioxide a tree will take up from the atmosphere. And it also tells us how fast a plant will grow. And so we can use these rules to kind of scale up and to not only study one given tree in the Amazon basin, but then if we know something about how many trees there are in the basin, we can take Leonardo's rule and we can scale that up to say something about how much carbon dioxide the entire Amazon rainforest will take in, all by knowing Leonardo's rule, okay? So to give you just a little bit of foreshadowing in what we're going to be doing today is we're going to be taking different forms of biology, but in particular trees, and kind of breaking trees down to their mathematized selves, okay? So here we have a perfect symmetrical kind of branching tree as we go from one mother branch, then to these daughter branches, then ramify all the way to the tips. And we're gonna make some assumptions about the tree. And we're gonna be measuring the radius and the lengths then of trees. And then we're gonna be paying attention to some of these branching rules, okay? That's just foreshadowing. Okay, but enough of these specifics. Let's get back to the main story. Okay, so before we get to trees, we need to see something a little bit more about how we've learned to see the geometry of nature. Okay, and so the geometry of nature, when you're taught geometry in particular in school, you learn Euclidean geometry, right? You know, so you know something about triangles, Pythagorean theorem, right? You know something about circles and squares. And from simple Euclidean geometry, if you remember back to your introductory geology classes, remember kind of Euclid's postulates, and you start with these different postulates, and then that kind of proves the existence of things like squares and circles and triangles. And then we can learn all these Euclidean rules about calculating the area of a triangle or figuring out the missing angle of a triangle and so on, right? This is Euclidean geometry. And Euclidean geometry is great because it basically starts with three basic dimensions about the world, right? Kind of the first dimension, a second dimension, and a third dimension. So the first dimension, Euclidean geometry, is simple. It's just a line, right? A line is defined by two different points, and it has a length, all right? So that's a one-dimensional object, a line, one dimension, described by its length. But we can then also characterize different shapes, Euclidean shapes, squares, triangles, circles, and we can characterize them with their areas. So these are two-dimensional objects, right? Something that we can draw on a piece of paper. And so what's nice is that an area can be distilled down to basically a length dimension. So areas in general are proportional to a length measure squared. Okay? But we can also go to another dimension, a third dimension, right? It's the dimension that we're familiar with. This is what we live in, 3 dimensions. And you can take these Euclidean objects, spheres, cubes, cylinders, and whatnot, and we can calculate their volumes in the 3rd dimension. And a volume is just a linear measure cubed, right? This is just basic Euclidean geometry. So we can go from lines to areas and volumes. 1 dimension, 2 dimension, 3 dimensions, okay? So we're taught that we exist in 3 dimensions, okay? And thanks to Euclid, this seems very rigid and natural, right? Nature is Euclidean. But the reality is that if you actually look at nature, it doesn't look like a nice sphere, a nice rectangle, or a cylinder. Nature looks like this. In fact, what I'm showing you here, this is the Amazon basin. Remember that picture of the Amazon I showed you with all those trees? But this is taken from a remote sensing platform, okay, usually with either an airplane or a satellite. And this is a laser scan of the Amazon where they're able to strip out the vegetation and look at the elevational differences of the Amazon basin. And what you'll see is this meandering river and all the different pathways that the river used to take, but no longer takes because it's been meandering then through the Amazon basin. Then you'll see these little hills. And this is a real reflection of what the geometry of natural forms look like. In fact, it's very rare in nature to find Euclidean objects, either straight lines, perfect areas, or 3-dimensional Euclidean geometric objects. These are very rare in nature. What's common are forms that look like this, or this. This is an enhanced photograph where we're looking at the water content in trees, but you'll see that the water content in trees ranging from a lot of water within a given tree that is a healthy tree in the blue to a stressed tree in the red, kind of takes on the form of the natural landscape, right? So this geometry in terms of landscapes looks very different than a Euclidean object, right? So when we look out on the real world, we see a different type of geometry, one that's not Euclidean, but something different. Artists, natural historians, mathematicians have struggled to deal with the geometry of nature since basically the time of Euclid. And if we look within biology, the forms that we have from biology are also very different than simple Euclidean objects, right? So to follow kind of our signature of artists leading the way, this is one of my kind of favorite paintings of Katsushika Hokusai, who was an artist in Japan. Okay, he lived between 1760 and 1849. And in this painting of the Great Wave is appealing on many ways because it kind of captures some very interesting attributes of nature. And it has actually captured the attention of many different people because actually if you stare at the painting of the Great Wave long enough, you'll start to notice some additional patterns. And sometimes what you'll find is that, well, geez, that's actually not a wave in the background. That's actually Mount Fuji, a mountain with some snow on the top, which kind of starts to resemble these waves and the foam on the top. So there's some similarities there. Others have noticed that there's actually some regular geometric shapes kind of superimposed on top, but kind of taken at different scales of measurement. So you can start to extract out some mathematical regularities that seem to hint at geometry. But there's also this repeating pattern. So if you look at the repeating patterns, there's this huge circle and these smaller and smaller circles that all of these different patterns then can repeat, as well as different patterns of triangles. So the more you look at the Great Wave, the more you see. Relatively recently, people have noticed that actually what he was painting was something that is now known as fractals, well before anyone coined the term fractal. So he noticed the fractal characteristics of nature. And this kind of the subset then here, this is known as a Mandelbrot function. This is a repeatable kind of regular pattern, something known as a fractal that we'll talk to in a little bit. So this is in the 1800s. Artists are seeing these patterns then within the world. So fractal geometry is about finding order in what initially appears to be chaotic or really complex. It's distilling out the simplicity from what appears at first look to be complicated. Okay, so I want to define for you a few terms. Okay, in particular, a term that we often use, something known as self-similarity. Okay, and so in mathematics, a self-similar object is an object that's exactly or approximately similar to a part of itself. That is, the whole of the object has some shape then that's reflected in one or more of the parts of that object. That is, there's some aspect of the parts that are repeated so that the whole begins to approximate the parts. And so many objects in the real world, such as trees, rivers, coastlines, are self-similar. That is, parts of them show the same properties at larger or smaller scales. So self-similarity comes with this notion of scale invariance. And so I hope I don't lose anyone with these terms, but I wanted to define these, and I'm going to give you some visual examples of that. But self-similarity is important because this is a typical property of fractals. Okay. All right, so all fractals show a degree of what's called self-similarity. And so if you look closer and closer into details of a fractal, you'll actually see a replica then of the whole. So for example, if you look at this fern, right? So if you look at the entire frond then of the fern, you're going to see these different branches coming out from the main stem. Okay, so hopefully you can see that. Now, if you zoom in then on those branches, each of those branches then looks similar to the entire frond then itself, but it's just at a smaller scale. And you'll see this pattern then that repeats and repeats and repeats. Okay. That's self-similarity. So self-similar patterns are the result of a simple equation. You can think of that as a rule, all right, or a simple mathematical statement. And the fractal itself is created by repeating this rule or this equation through a feedback loop. Okay, we call that iteration. So we're going to iterate this rule over and over again, the same rule repeated. Okay, where the results of one iteration are then form the input value for the next iteration. Okay. So it's a way in nature to create very complicated structures from very simple rules. Okay, so if you look at this Nautilus shell, what you'll find is that each of these different chambers are just a scaled down version of its neighbor, and it's just repeated over and over again. Okay. And so most of us then, if you take basic geometry, are taught that length, width, and height are characterized by these three discrete dimensions of a linear dimension, areas, and then volumes, right? Now, fractal geometry, which is basically the real geometry of nature, kind of throws this concept kind of a curve. Our typical 3 dimensions now are going to, instead of being these discrete 1, 2, and 3 dimensions, are now going to be continuous. That is, we can have dimensions that run between 1 and 2 dimensions, between 2 and 3 dimensions, and even between 3 and 4 dimensions on up. And the fractal dimension of a shape is a way of measuring its complexity. Okay. All right, so within math, the person who really kind of not only understood the importance of the fractal geometry nature, but the one that basically coined them the term and formalized this in a way that we can use it is a person by the name of Mandelbrot. And in 1975, he basically kind of formulated a lot of these ideas. And so this is a great quote from Mandelbrot. And he says, clouds are not spheres, mountains are not cones, coastlines are not circles, and bark is not smooth, nor does lightning travel in a straight line. So Mandelbrot introduced what he called the fractal geometry of nature, and because of which we now have different sets of geometry. We can describe the world in terms of Euclidean geometry, spheres and cubes and cylinders, but also fractal geometry. And the key idea with fractals is self-similarity. And so an object that's self-similar can be decomposed into smaller copies of itself. But then the property of the whole, that is the fractal, is contained in the rules then that make up the parts. Right? So in stepping back, what the fractal geometry in nature is telling us is that very complicated structures can be created by the iteration of very simple rules of nature. And so Mandelbrot basically gave us a whole new way of looking at nature, not as one of a complicated mess going on in the real world, but instead a wonderful complexity generated by underlying simple rules. All right, so fractals are the result of a construction procedure, an algorithm that's recursive or iterative, that's repeated over and over and over again. So it's this constant iterative rule that then generates the patterns, the complexity that we see around us. Okay. So the famous one is something known as the Mandelbrot set, right? So this is your kind of typical fractal, okay? If you've kind of Google fractal, you'll come up with something that looks like this, usually the Mandelbrot set. Very complicated structure. But this structure is just a simple function. And that simple function is iterated at each of these different scales over and over again. And it just produces all of this incredible pattern, right? So we have now these different objects of nature. We have fractal or self-similar objects, right? And we have Euclidean objects. And so I think in the interest of time, what I'm going to do so that we're able to kind of cover everything is I want to kind of go over, we'll do one kind of little lesson here, okay? And that is, how do you actually quantify a fractal in nature, okay? And it has to do with measuring something known as a fractal dimension, okay? And so hopefully this doesn't scare you, right? So usually when I mention fractal dimension, be like, what? You know, what is that? But it's actually, it's beautiful in terms of how it's calculated, and it's quite insightful. And I'm going to tell you how we can use fractal dimensions, measuring fractal dimensions, to actually pick out. This is one of the methods that people use to identify whether or not a painting is a fraud or not, okay? So bear with me here. All right, so a fractal dimension is a mathematical description of a self-similar property. So remember, to be a fractal object, one has the property of being self-similar, okay? Now there's many different ways to characterize a fractal dimension. I'm gonna show you one way, okay? And it kind of boils down to this simple little equation, okay? And it's just a quotient. We're just dividing then through here by something. Now think of any kind of object. So let's start with a line, okay, a one-dimensional line. Now you can subdivide that line, okay, into a bunch of n identical parts. So if we cut it then in half, we'll have two n equals 2 parts. If we cut it in 3, we'll have n equals 3 parts. And each of those parts is scaled down by a factor. That is, as we keep kind of cutting, you know, our parts are going to get smaller and smaller and smaller, right? And so the size then of those parts as we keep cutting is just 1 divided then by the number of cuts then that we make, right? So you can think of as this value of R as that's our minimum resolution as we zoom then into the line, right? So we're going to do kind of the fractal thing and zoom into the line, right? Based on this scale factor R. All right. Now, it turns out that a line is one-dimensional, as we told you, okay? An area is two-dimensional, and a three-dimensional object obviously is in 3 dimensions. Now, each one of these different objects can be divided into n different parts, right? And you can show kind of mathematically that for a line, two-dimensional objects or three-dimensional objects right? That this formula here has to be obeyed, right? So we'll know as we keep cutting up these objects in the smaller and smaller bits, we can calculate then the size of that bit based on whether or not we're doing it in 1, 2, or 3 dimensions. Okay, so this is kind of a little bit of math formality. But now what's really interesting is that it turns out that for a self-similar, for a fractal object, that same equation holds, but instead of having a number either going from 1-dimensional, 2-dimension, the 2 is from 2 dimensions, 3 is from 3 dimensions, now we have D here instead. And it turns out that D, if you solve for what D is for any object, that is the fractal dimension. That'll tell you how fractal then that object is, okay? And so don't get scared by logarithms, but another way that you can find then D is then to take the logarithm of both sides. Don't worry about that so much. But I can show you now what's really interesting and unique about fractal entities and how life, living things, the natural world differs. So for a self-similar object, D is then our measure of fractalness, how fractal it is. So remember, for a line, the dimension is 1, but you can make a line more and more fractal. That is, think of your line, and now let's start moving that line around. All right, so if you're drawing the same length of a line on a piece of paper, a straight line, its dimension would be 1. But now let's take that line and start zigzagging it, okay? Or spiraling it around, right? And if you do that, you can take a line, you can start to color in almost a perfect area of a square or a circle. So when you color in then a shape on a piece of paper, you're taking a line that's 1 dimension and you're making it look like and approximate the 2nd dimension or an area. Same thing for a surface, a 2-dimensional surface. If you crumple it up, all right, you can start to make a 2D surface into 3 dimensions. The same thing for volumes. And this is where biology comes into play. And it actually turns out that within biology, biology takes 1, 2, and 3-dimensional things, and natural selection has kicked it up another dimension. And we're going to talk a little bit more about that because that may seem totally esoteric. You'd be like, what? Biology makes things into a 4th dimension? So it turns out that life is not living in 3 dimensions, but actually lives in 4 dimensions. Now bear with me. The 4 dimensions actually comes from these basic kind of Euclidean dimensions such as lengths, areas, and volumes. But if we actually measure these different biological attributes actually measure biological volumes. Let's use the volume of your lung, the volume of the canopy of leaves then within a forest, or the rooting volume. They don't follow these simple Euclidean rules of just simple, for 1, 2, or 3 dimensions. Instead, every time we measure biological lengths or areas or volumes, they always tend to be 1 dimension higher. So that volumes ultimately, instead of acting as a linear dimension cubed, they act like a linear dimension to the 4th power. And at all of these different length, area, and volume scales, biology actually is different, okay? Because natural selection has pushed surfaces, volumes, and lengths to be fractal, okay? So one way I can get you to think of this, okay? So within your body yourself, you have an amazing lung and you have a gut. Your lung and your gut is made up of surface areas, okay? Like a piece of paper. All right, so if I spread out your surface area of your lung, it would actually fill up most of the room that you're in right now. In fact, a good chunk of your house in terms of the total surface area. But what Mother Nature has done in natural selection is that it's taken all that surface area by which you absorb oxygen from the air that you breathe and scrunched it down into this highly convoluted structure known as the lung. So your lung really is a two-dimensional object of surface areas over which you exchange oxygen from the atmosphere and then transport it into your body, okay? That's what the lung does. Same thing with your gut. When you eat food, your gut absorbs through the surface area of your gut. But your lung or your gut is a two-dimensional area, but it's packed and folded inside your body in a very wonderful, kind of complicated way that maximizes surface area within your body. And so here, if we take a fractal dimension of a piece of paper, it's 2. But as we proceed to take that two-dimensional area and make it more 3 dimensions, right? That two-dimensional area is now starting to approximate a sphere, which is a fractal dimension of 3, but it's not quite perfect. So when we measure the surface area of a lung or a gut within your body, we actually get a fractal dimension coming out higher than a pure surface. In fact, that surface now starts to approximate a volume, okay? A two-dimensional piece of paper can start to approximate a sphere if you crumple it up in a confined space, okay? So this is somewhat complicated, but it's kind of obvious, right? Now, how do we calculate the fractalness of something in nature? So this is something known as a box counting method, okay? And we're going to be doing something similar to this when we start drawing trees. So let's imagine we have an image of a branch or a root or something, right? And it's kind of pixelated here. So this is like a picture or something. And you zoom in, you can see all the different pixels here. Now, what we can do with our image is that we can slice it up into different resolutions. And so this is the full resolution, or we can slice it up. Then we can cut these in half, okay? And so we can slice it up into finer and finer resolution. And what we do when we measure and we want to measure how fractal something is, at each of these different resolutions, we count how many times this image or this root is observed. So in this one big box, or our first kind of pixel here, we find that, yes, we have one image of a plant inside. Now, if we start kind of subdividing this further and further, what you'll actually find, there are 4 squares, okay, or 4 kind of pixels, but we only find 1, 2, 3, or n equals 3 pixels with a plant in it, okay, or a branch in it. And we can keep doing that. So here we have 11. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 boxes where we see the plant, right? And so notice there's some gaps here. And as we keep subdividing and subdividing now, we're going to be counting these gaps as well as boxes then that are occupied. This is known as box counting, a way of measuring then the fractal dimension. And so we count then all of our different boxes. We keep track of how many have plant images and how many don't. And then what we do is we plot the resolution of our measurement versus the number of boxes that contained our plant as a function then of the resolution of our analysis. And we just fit a straight line to this. And notice this has to be done on a log-log plot because that's based on the definition then of fractality. And notice the slope of this relationship is 1.6. Actually, it's not minus 1.6, but the fractal dimension of this is 1.6. Now notice that we are looking at, this could be a series of different lines. It's a one-dimensional object, but it's trying to fill a two-dimensional area, right? If this was a straight line, the slope of this relationship would be 1. That is, it's a Euclidean dimension of a line, which is 1. But this is not a line. This is a complicated structure. But it's not a full area either. That is, boxes don't fill the entire area, which would give a slope of 2 or 2 dimensions. Instead, this structure fills a dimension between a line and between then a totally filled area. Its dimension is 1.6. And so we say that this object has a fractal dimension of 1.6. Now notice the self-similarity here. We're analyzing then the structure at these different resolutions or these different scales. And we're looking at a pattern that is the the number of plant objects we encounter. And if we see a straight line on a log-log plot and we get a slope of that relationship, that tells us what we're looking at is a fractal, okay? So that's an easy way that you can not only measure the fractality of something, but also tell if it's a fractal or not, okay? And what dimension that object fills, okay? So we can use this method, and this is one of the methods we use to characterize patterns in nature. So for example, this is a distribution of, this is an inverse image of city lights. I think this is from a section of Brazil or something. And you can study patterns of human occupation across the landscape by calculating then the fractal dimension of city occupations or human occupation across the landscape. We can also use imaging to look at the presence or absence of tumors or cancer. So normal tissue has a general regular pattern, but a cancerous tissue would then result in a totally different pattern of fractality. Okay? So we can use this as a measure of a healthy or a potentially a sick individual. And so this method of box counting is quite useful because we're using this iterative notion of looking at patterns at different scales to help us describe then nature. Okay. And we can do this in several different ways. And there's these different algorithms then that measure what we're looking at different scales, and we can plot out these relationships, and it tells us something about how the world works. How does this relate to art? Well, it turns out that artists kind of have sensed this notion of pattern occurring at different scales. Drawing patterns at small scales, keeping that same kind of iterative rule as you then proceed at different scales. Okay. So if you're not familiar with, actually one of my favorite artists, this is Jackson Pollock, right? And so at first, I think I took my kids to see some Pollock paintings in New York. And at first they're like, well, I can do that, right? That's easy. I just throw my paint at the wall and there you go, you have a Pollock painting. But he had a specific rhyme and reason and a method for doing this. And actually you can compare my kids' patterns of paint on the wall to a Pollock painting, and you can immediately tell by doing this box counting or this fractality measure, you can extract out the unique signature then of an artist. So is this a fake or an original Pollock painting? So it turns out that if you analyze this box counting method where you kind of subdivide Pollock's paintings and you do different sorts of measurements of whether or not you see black paint or white paint or the intensity then of the number of paint drops per unit area, and then plot out the relationship then between the scale of the measurement and the number of observations, whatever that is, at different scales, you'll get a unique signature of Pollock, which is this line, and the slope of that relationship is Pollock's fractal dimension. So Pollock was painting in a fractal way, that same patterns at different scales. Okay? And it turns out that you can do this across nature. And you can extract out these very regular curves in terms of how vegetation or leaves or paintings then are described. And they're usually very unique in terms of their fractality. So what's amazing to me is that you can take something that's so complicated and make it a simple equation for a straight line. Okay. That simple equation for a straight line ultimately reflects underlying rules of how the geometry of things come to be. So I'm going to end here before we get started with kind of what we've kind of discovered about the morphology then of life in terms of biology. And in particular, we've been fascinated by these vascular networks in biology, right? And so here you are, maybe dozing off, listening to me, strip away your skin, you are an incredible network, okay, underneath. And so this is your vein and arterial network that supplies oxygenated blood, right, to all your cells, and then takes then the deoxygenated blood, okay, and moves it back to the heart, where it's then moved to the lungs to pump out again to all of your cells. You are a living, breathing network, right? And plants are no different, right? So if you take then a leaf and kind of zoom in at increasingly larger and larger scales, or smaller and smaller scales, depending on how you want to go, what you'll see is this amazing network, this structure of this fractal-like venation network, okay? Your lung, okay, a bronchial lung, transporting oxygen and CO2, okay? So you're taking then this volume and trying to fully pack in as much surface area then within your lung, okay? All your nerve networks look like this too, right? These tree-like networks where we have these terminal branches all kind of filtering down to these main stems, right? So animals and plants, okay, here are some invertebrates, okay, these wonderful corals, these filter feeder then networks, right? A very similar theme, right? Here is a fossil organism that lived, oh my goodness, probably this is the Burgess Shale. This is about 500 million years ago, maybe 600 million years ago. We see these evidence that these forms of life have existed for quite a long time. So tree-like networks, right, are kind of everywhere. Well, why do we see tree-like networks in biology? Well, they're fractal, they're self-similar. It's this repeating pattern of these branching rules. And the argument is that evolution by natural selection has operated across all organisms to kind of shape then their delivery networks, these branching networks, according to a few different principles that are basically the same across animals and plants and appear to have not changed since the beginning of life. And that is, a lot of these networks tend to be space filling. That is, they try to kind of fill the entire body, of the organism, such as your lung or your gut, or the venation network tries to fill the entire leaf. That is, they try to fill the space, right? But also we know from Leonardo that these networks are actually area preserving. That is, as you go from mother branch to daughter branch, as the cross-sectional areas are preserved, as Leonardo told us. And there's a few different reasons for this. One is biomechanics, but the other is having to do with minimizing the energetic throughput of the network. And the end result is that these networks have been shaped by natural selection, it appears, so that they transport oxygen or water or nutrients to maximally supply all your cells so that all your cells are as happy as possible, but do it at a minimum cost. It's kind of like the most bang for the buck, right? You want a structure that costs the least, but gives you the most supply. Okay? And that appears to be what natural selection has hit upon. And so you can draw lungs, you can draw your arterial network, you can draw leaf networks or trees according to some pretty simple rule. So these biological networks originate then from simple iterative branching rules. They're fractal, they're self-similar. If you look at any one section, okay, and understand the branching and you zoom in on another section, the branching is similar, but they follow iterative rules. And the diversity of life is really quite astounding because nature has created all these different forms from just a few small, simple number of rules. Okay. So why do biological networks look so similar? Well, the argument is that evolution by natural selection has shaped the geometry of these biological distribution networks really to follow two principles. In general, they tend to be space filling. That is, they kind of fill some sort of space, all right? So the root network kind of fills a rooting volume. The branching network above fills this canopy volume. Same with your lung, right? And so the space filling idea is that another way you can think of it is that as you go from the mother branch to the daughter branches, the volumes, if you sum up the volumes of the daughter branches, the volumes then equal the volume of the mother branch, right? And the same thing for the area preserving. That is, if you look at the cross-sectional area of the mother, that cross-sectional area is preserved as you go out then to the daughter branches. And so the argument is that on the one hand, these networks try to maximize resource exchange surfaces, to exchange resources from the environment, be that carbon dioxide in the atmosphere or oxygen then in the atmosphere, but then at the same time try to maximally supply it and transport it at a minimum cost. Okay. So we can simplify nature as this wonderful, nice, symmetrical branching tree where we have our mother branch and our daughter branch. And so this is getting into our art section here. So notice on the mother branch, we can characterize the radius then, okay, how thick then the branch is, but also its length. But then as we go from mother branch to daughter branch, we can characterize the daughter branch also with links. But notice here what we do, we have these little, this notation here we call L sub K and R sub K. So R is radius, L is length. K is just the branching level, right? So you can go from our basal branching level, level K, but then we can go to K plus 1, right? That's one branching level up, or K plus 2, K plus 3, K plus 4, right? Each kind of iteration. So notice what happens here at each iteration. This is the iteration. This is where the fractal comes in. We're now going to have a rule that we're going to invoke in terms of how the radius and the links then are going to be following a constant rule as we go from one branch to the other, to the next and the next, and so on. That's the iteration. And we're going to keep these branching rules kind of steady as we then go out. So we're going to make some assumptions, right? As we go from the trunk then of the tree, be that the aorta, okay, or the trunk of a plant all the way up to the tips, okay? So we're going to assume that the tips then are always kind of constant, and we're going to measure some lengths and radius and so on. So don't worry about some of this detail, okay? But what I want to kind of show you here, and ignore the math a little bit if this scares you, but I just want to focus on this one kind of branching rule. We're going to call this branching rule number 1. As we go from level K to level K plus 1, the volume then of level K is the number of branches of level K multiplied by the length cubed. So remember, the length cubed is the volume, okay? So we're going to kind of assume that there's another kind of constant here that we're going to ignore, having to do with this is a cylinder, right? We're going to make this mathematically simple. So the volume then of this main trunk, we're going to assume it's a cylinder, is just its length cubed, okay? It's going to be proportional to its length cubed. Now, the volume then of the next level, K plus 1, is the length cubed of the daughter branch times the number of branches, which is 2. So notice we're going to enforce that the volume then at this level is going to be the same as the volume of this level, okay? And so as we then go out then the tree, if we add up all the volumes of the branches here at the tip, it's going to equal then the volume then of the mother branch down below, okay? And that is known as volume preserving branches. That's the rule. So as we go from one branch to the next, we're going to, Mother Nature has enforced approximately this volume preserving rule, okay? And that you can see that in terms of how the links then change as you then go through the network. And so we made this simple. This is a symmetrical tree. And so the next rule that we're going to impose on our biological trees is the Da Vinci or Leonardo's rule, right? Area preserving. And in area preserving, the rule is we're going to take the cross-sectional area, all right, of the trunk, which is radius squared, multiplied by the total number, and at the base of the tree, it's 1, right? Now, the cross-sectional area of the next generation is the cross-sectional area of the daughter branch squared times the total number of branches, which is 2. So that has to equal each other, okay? That's our self-similar or a fractal rule. And then that's repeated over and over and over again, okay, through then the network. Now, we can then plug that in, and there's a lot of fancy math here, but we can then calculate a lot of attributes then of the tree, such as the total volume of the tree, how big it is, right, how many leaves it has. And if you'll notice that when you do this, we often deal with these exponents or these powers. And that's why we do log transformations back and forth, because when we iterate this, there's a power often involved. So I'm going to give you one more third rule. And this is mathematically a little complicated, but I can make this a little simple. So it actually turns out that real trees in nature are fun to draw, and even Leonardo noticed this. You can start drawing trees by assuming a symmetrical tree. That is, where the daughter branches are always the same lengths and radii, right? But it turns out in the real world, perfectly symmetrical trees are really rare. In fact, we very rarely see symmetrical trees in nature. Instead, we see asymmetrical trees. That is, as we go from mother branch to daughter branch, often one daughter is bigger than the other in some way. And there are these different types of asymmetries. But it turns out that if you look at the relative proportions then of the two different daughter branches, the volume preserving and the area preserving can still be enforced. That is, the two branches of the daughters then can differ, but they can still ultimately be constrained. Their sizes are constrained by rules number 1 and 2. All right, so the third branching rule is we're going to allow then the daughters to be asymmetrical, but that asymmetry is not free to vary. That is, they still, when you add up the cross-sectional areas of the daughters and the volumes of the daughters, they still have to equal or approximate the cross-sectional area and the volume of the mother, okay? But once we allow then this third rule then to happen, it actually turns out that between just those 3 branching rules, right, you can create trees that look like, oh my goodness, a lot of what we actually see out in the real world, right? So here, all the way on the left, this kind of looks like maybe, this is like a bujum tree. So those of you who've seen, been down to Baja to see some of the bujum trees, or an acatillo, right? Maybe one branch of an acatillo, maybe some maple or some oak trees, all the way to a perfectly symmetrical tree. So between those 3 rules, okay, if we keep the volume preserving, area preserving, but then we allow then for some asymmetry depending on the local environment, you can create a bunch of different, what look like real world trees, okay, with 3 simple rules. And again, this is amazing to me because just with 3 simple rules, you can create this tremendous diversity, right? And it turns out that we can map those different types of branching architectures into different environments. So in environments where light is really limiting, like in tropical forests, you tend to get trees that start to look like really symmetrical trees. Whereas in areas where maybe light, you want to minimize your surface area to avoid kind of light, but still have maximizing kind of your hydraulic capacity then of the network, right? In desert type environments, we tend to see structures that start to look like this, right? So 3 different rules, then we can characterize the nature that we see. Okay, so to conclude. So I kind of started out kind of showing you kind of the history of how both art and science have kind of evolved then together, right? Often with artists then kind of showing the way of how to kind of make sense of the world and to see kind of the geometry and nature of new light. And then the mathematicians and the biologists and the natural scientists kind of coming in later, picking up on these ideas and formalizing it in the language of mathematics, right? So I gave you an introduction to self-similarities and fractals, and I showed you one method so you can actually go out and you can measure the fractalness of an image or whatever you're interested in. In fact, there's some nice tools online where you can do that box counting method yourself. And then I kind of ended here with some of these rules of life, right? And so when we look at these branching networks in biology, we can actually extract out these different rules of life and show that there's this remarkable kind of unity and across organisms in terms of these branching rules, in terms of how they're kind of organized. Okay, so with that, if you're really interested in any of this, I'm giving you a very kind of short kind of overview. I would highly recommend these books. In particular, especially Mandelbrot's book on the fractal geometry of nature. It's a wonderful coffee table-like book, and it's a great introduction to some of these ideas. I didn't have time to talk about Ernst Haeckel, who is this early kind of biological artist, as well as a great scientist who kind of also kind of discovered some of these different rules by which you draw then nature and kind of express nature. And Leonardo da Vinci's notebooks, you can now buy this in these nice kind of bound kind of copies for pretty cheap. And if you're interested more in Mandelbrot, we actually did a PBS Nova series, which you can watch online. It's called Hunting the Hidden Dimension. Unfortunately, Mandelbrot passed away a few years back. And it's a shame that he never received the Nobel Prize for his work, because I think the longer time goes on, the more people have realized that his insights and his way of kind of mathematizing kind of all these concepts has really impacted and led to amazing insights and innovations, not only in biology, but also in physical sciences and also in engineering and a lot of practical applications too. So I would go to those places if you want to learn more. And so then with that, I think I'll end. And hopefully I didn't lose too many people.
No.
All right.
You could stop your screen share there, Brian.
Sure. There you go. That
was truly amazing. Whose mind's blown? I mean, mine is like, boom. And I'm bringing Paul back on too here. Great. Wow, wow, wow. That was truly fabulous. And now it's just going to get even more fun. Paul's going to take it from there, and you're going to get to draw these. If you draw along with Paul. Paul, I don't think we have time to do the first little exercise. Let's get right to doing the Leonardo tree, because we want to give you plenty of time for that.
Yeah. Just listening, Brian, I'm so glad you brought in Jackson Pollock, because really briefly, the most common tree around Tumamoc Hill in the desert here is the Palo Verde tree. I started a photography project photographing these Palo Verde trees, and I noticed that they started looking kind of like Jackson Pollock paintings, all of the complex. I was going after maximum complexity in these photographs of nurse trees. And I found this paper, but I can't, apparently Jackson Pollock, people wondered why he just kept doing the same, like throwing paint around like for 20 years. When you measure those paintings over the course of his career, they became more and more fractal. Are you aware of that paper?
Yes. Yeah.
So they weren't just random. He was getting the feel of this natural flow of the paint can that was on a string that was going around in these kind of really natural fluid motions that was producing the paint. So it wasn't like just throwing at random. I got very interested in measuring some of the fractal dimensions of these Palo Verde trees. So what I'm going to draw here is going to be influenced a little bit by all this work that I've been doing on Tumamoc Hill as part of my artwork. So it's really wonderful how you blended that in. What I'm going to start with is the motto, my motto, and the sort of the basic rule of the Tumamoc art and science, which is you don't really understand something fully until you draw it. So all of this math kind of stuff, my brain sort of fuzzed out a little bit, Brian, but when I've drawn these things out, I started to understand them. So I'm going to turn on, cue me now on how I turn on my
Yeah, so your first share, let's go to share screen and then your advanced tab and select the document camera.
There we go.
Second camera. Perfect. That's showing the notebook.
So,
and folks, if you want to sketch along with Paul, those of you who printed out the cool fractal protractor Paul created.
I cut out of a piece of manila folder. I'm drawing, I've worked out some math here, and it's fitting on a landscape piece of printer paper. So first of all, I'm going to start with the basic little seed element of the fractal that's producing trees. I'm going to simplify and draw out some of the things that Brian was drawing. So to get at Leonardo's rule, so that everything here is a cylinder. This is the mother branch, and then these two daughter branches here. I find that drawing things out freehand is where I learn the most. Sometimes I don't really use rulers so much for this because it's faster, but also drawing freehand, your body starts to understand these things, not just your brain. So there's these, you can look at these as cylinders hooked together, and I just draw this out so I can understand Leonardo's rule. These cross sections of these cylinders, I'm going to call this D, big D, diameter 1, and I'm going to call these little d, these diameters. So I went around the office here and I measured different Palo Verde trees, and I found that they all defied, they were all different, but they all converged pretty much when you measure a lot of them. They kind of converge on these particular proportions that Brian talked about and that I calculated here according to Leonardo's rule, which I'm gonna write with a thicker pen here, so that you can write in a more simple. The simplest way you can write this rule is areas are always a square. So squares are all everywhere in nature. D, which is the big diameter of the mother branch here, equals D plus D squared. So this is D1 and D2. If this one here is daughter 1, daughter 2. So we're looking at these cross sections of these cylinders, and till I drew it out, I didn't understand this formula. But of course, these two things, the two daughter areas here, equal this area, and it's easy to see once you draw it out. In fact, my daughter, who's in math graduate school, helped me break this Leonardo's formula down, and I'm going to quickly do it. You can do d squared equals, of course, 2 times the d squared here. 2 of the daughter squared is the same thing. You can break that down further into doing some algebra, which I haven't done since grade school. You cancel all this stuff out, and you get the square of the big diameter over 2 is the square of one of the smaller daughter diameters. And if you further break that down, as she explained it to me, you get the square root of 2, which is really, really interesting. Now, the square root of 2 is if we're going to call this 1, that's 1 over the square root of 2 equals like 0.7. So there's something about the ratio of 0.7 in nature, because the square root of 2 is sort of like we went through in the Fibonacci numbers a couple weeks ago. There's a ratio of certain ratios that appear in nature, and the square root of 2 is one of those. And I asked her, like, that's just weird. Why the square root of 2? There's a couple things. Partly because area is related to squares. And a lot of things are just related to 2. So you get a lot of things that are 2 in the square root of 2. One of the things about the square root of 2 is that it's an irrational number. It's 1.414, and it goes on infinitely. So nature seems to use these irrational numbers a lot to create something that never quite adds up. So you get this wonderful kind of indeterminacy or randomness, because the square root of 2 is the irrational number. And let's just use this. I'm going to use this 0.7 as a factor to, so if we're using this basic seed thing here, this basic seed shape that's going to form a tree, I found that if you multiply the diameter of the mother branch times 0.7, you get the diameter of one of these daughter branches. Like for instance, this would be 0.7. And so when looking at these trees, I took some photos and measurements with a protractor. I don't think I have time to show them, but at first I thought each of these daughter branches should be like half the diameter of the mother branch. But obviously, once I understood area is a square, not just a particular half. So pretty much these daughter branches were a little thicker than I thought. Like I thought if they conserve area, they would be, half the diameter. And I walked around with a ruler. It's just something that after doing these calculations, I noticed that now I will notice a lot more. And let's just get our big piece of paper and just start drawing this, and it will make sense. I'm going to start out with the trunk. Let me adjust this so that
and Paul, if folks are drawing along, they're going to want to start their trunk way down at the bottom of a horizontal page.
Way at the bottom, I've kind of calculated this out that the square root of 2 actually is something that's found within these trees, but it also fits on a piece of printer paper, which I have a pile of here, which is easy to draw on. So I made this little protractor. I also measured a lot of these trees, and I found that 60 degrees was a really, a lot of them were like 10 degrees off, larger or smaller. But I chose 60 degrees as kind of an average. So there's 3, so let's just take 2. There's this little seed element that we just talked about. I'm gonna take this and I'm gonna vary it, first of all, with the same angle so that it has a self-similarity, because this is always going to be 60 degrees. And then I'm going to vary the width, which is the diameter of these branches, by 0.7, so that you can kind of intuitively understand that if there's water and nutrients flowing through this branch, you don't want to have a blockage when you come to these daughter branches. This same number, this is flowing all the time. The same amount of volume always has to be flowing out into these into all these other branches, and it's intuitive just thinking about it that way when you draw it out. So I'm going to keep I'm going to keep this angle constant to keep the self-similarity. I made a little scale on here. You can see I started off with a 2-inch mother branch, and I multiplied everything by the square root of 2, which is 0.7, and I got kind of a descending scale where the next one is 1.4, 0.98, 0.6, 0.48. I just made a little square root of 2 proportional ruler here. So I'm going to make the first set of branches. And like, how am I going to calculate how thick this is? So if I measure, I think I'm not going to deal with thickness at this point. I'm going to just estimate the thickness. But these daughter branches will be 0.7 if this is 1. And so this is the first set of little cylinders. Here is going to be just forget about these. So this is going to be 2 inches here. The next one, we're going to keep this volume, we're going to keep this angle constant. And these branches, again, by 0.7, multiply the mother branch by 0.7, these will be just a little bit bigger than half that diameter. And if I had the time, I would measure both. I would measure both of these with a ruler. But according to this, if this one is 2 inches, if this one is 2 units, the next one here is going to be 1.4. So that's the
that is, Paul, because you multiply by 0.7.
Yeah, so I'm multiplying the mother branch here, which is 1. This one is actually just arbitrary. This is just the trunk. I'm considering this the mother branch, which is 2 inches. If you multiply that by 0.7, you get the 1.4. So this is my iterative formula, which is found in trees. And I'm not saying that this is the formula for a Palo Verde tree, but I'm taking this same little Y-shaped seed element and I'm applying the same angle and the just estimating the equal area, and I'm multiplying the length times the square root of 2 proportion, which is 0.07. So 2 times 0.7 is 1.4. And I'm just going to keep on doing this. And the branches will keep getting shorter and shorter and as they get more branched. And it's also intuitively logical to me that for structure, besides fluid movement through the branches, as a branch gets thinner, it's weaker. From an engineering point of view, as you have wind and things blowing through trees, you want to get brancher that's thinner, you want to get it shorter so that it has more strength to it. So this is 1.4 times 0.7. You're going to just get a little under an inch, 0.9. I'll just say about drawing. Drawing is just kind of a meditation, and you can draw these fractal trees as not as a math exercise, but just as sort of an exercise for the mind. And your brain just sort of enjoys dealing with these little spatial dimensions. It's a pleasurable thing. And it helps develop, in my opinion, visual, spatial kind of intelligence in your brain, just to do a lot of drawing. Doesn't matter what it looks like. And I don't care what this looks like either, but it's this thing, the mind just sort of like loves fractals, maybe because the brain is a fractal itself. But there's something really pleasurable about looking at those Palo Verde trees, and I couldn't figure out why these boring, really trees kind of started becoming more and more obsessive to me as far as my photography. And I'm still working on that, but this is, here's just our tree that we've got so far.
Paul,
And
what was the next measurement? So 1.4, your next one after 1.4 is what measurement?
so here, since I'm using this one as, this next one here is 0.9, so it's just a little less than 1. And then after that, we're going to go to using the same 60-degree angle. I got, a little over half of a unit, 0.6. And since I'm rushing a little bit here, I can go over this again, but I've got a little calculator on my phone, which I used to calculate this ruler, and you can use a calculator to arrive at these things. So everything's kind of, there's sort of this pattern starting to emerge that's based on this proportion of 1 over the square root of 2, or 1 over the square root of 2, which is 1.414 on to infinity and beyond infinity. So what have I got here? 0.6. So there's 2, 1.4, 0.9, 0.6. So the branch lengths are getting shorter and shorter as I apply this parameter to them. So and then as things start getting smaller, I might just eyeball all of this. Like I'm just sort of eyeballing the thicknesses of these things. But an easier way to do this is I'm going to measure with my little compass here. I'm going to get my little 1.6, my little 0.6 measured out, and to make things easier.
So if you're sketching along, you'll start to feel this. This is why we sketch these rules, is you start getting the feel of the fractal branching here and the relationships between the daughters and mothers. And eventually Paul and you, if you're sketching along, you're going to have a symmetrical tree appear on your page here. It's very hard to draw and talk at the same time, so I'm helping Paul out a little bit.
Yeah, go ahead and talk.
And Brian, if you want to jump in.
No, Brian, I just don't know. One thing I don't understand is
Sure.
how the lengths are calculated to make the volume. I'm not quite sure I understand how this conserved area determines what the length is.
Yes. So there are 2 branching rules that are governing both the radius and the length, but the radius and the length then are going to be constrained because the volumes then have to be preserved, right? So there's a few different ways that you can do it. One is that if you then set the Leonardo rule so that the cross-sectional areas are then preserved, that will then give the radius, right? But if you're drawing a symmetrical tree, right, and each of your branches, then you're drawing cylinders, basically. Each branch is a cylinder, right? So then, in order to invoke then the volume preserving for that radius, right, of a daughter branch, the two ultimate volumes of the daughter branches have to equal then the volume of the mother branch.
Yeah, and isn't that a function of the length of the branch?
That's right. That's a function of the length, right? So the volume then is going to be the length multiplied then by the radius squared. Yep. So that would be, yeah, proportionality. There'll be a constant in front of it, but it'll be volume is proportional to length then times the radius.
Okay, so that's one thing that I was wondering
Squared.
while doing this. Am
Yeah.
I conserving the volume here with this?
It's probably pretty close.
Because I saw that the areas conserved this square root of 2 relationship,
Yeah.
and so I just used the same one for the length.
Oh, this is great. I like your logarithmic protractor. That's awesome.
And
So one thing I just want to emphasize is that the structure that is going to be emerging here, is going to be, it looks complicated, right? It's it's a fantastic branching structure, but again, it's just emerging from these just simple rules that are iterative at each level. So what I like about this is that you're really emphasizing, the fractal nature then of the tree, going down to this rule of 0.7, right? So it's then that's then iterated. So you're always taking a constant then proportion, at the next level, right? And then that rule is kept the same at each of the different branching levels.
yeah, so right now I'm on, what if they I'm on the 5th generation, and that's 0.48.
Yeah.
So I started with 2 inches here.
Yep.
And I'm at the 5th generation, and they're just getting thinner, and they're starting to get more complicated here.
Yeah.
But just taking the simple rule of constant angle, and applying the length to each mother branch, applying that ratio of 0.7, which we calculated, which would keep the area the same on the diameters here. And I'm just using that same ratio for the length. And it's going to take a little extra time. So I'm gonna quickly I might use my, so I'm doing 48 now on this fifth generation. I know this is probably too fast to really follow along. But just to review this at home in your studio or dining room table, just to draw these things out is it's just kind of a pleasurable experience. And to me, drawing is doodling, any kind of like work with a pencil is increasing your neuroplasticity for spatial and what I would call creative kind of thinking. And so by drawing this, according to my theories, I'm understanding it both with my hands as well as my brain. So the brain can do all these calculations like Brian was showing, and it's proven with logic. But to understand it with both your hands, which is to me, the hands are part of the brain. They're connected, and they're like an extension of the brain. And they understand that the hands tactily understand this. Once you start drawing, if you don't really quite have it in your body. And this math or these like calculations become part of your knowledge, and when you've drawn it out, it also increases your understanding of it. But a lot of research has shown that when you draw something, you remember it maybe twice as long as if you're just looking at words or numbers like formulas. And it is kind of true, you can't talk and draw at the same time. And that's an interesting thing in itself, because you're using a different part of your brain. I'm using my kind of right hemisphere, my spatial brain, in order to do this, to figure out the calculations, and I may even make a mistake if I'm talking. Talking just uses a completely different part of your brain for the most part, which can kind of interfere with each other. So like sometimes, you know, you can't walk and talk at the same time because they're two different parts of your brain. So this last thing, this is, I'm getting down towards smaller and smaller, and I'm just going to probably wing it and do it by
So here's a question for Brian, maybe while Paul's just sort of finishing that out, because we should move to the next exercise shortly, Paul. So Kathy asks, so the measurement of the mother may change, question. Do we measure the mother length? And that is what we used in the formula. So in this case, this is a symmetrical tree. So the 0.7 is staying constant. But yeah. I think in the next example, Paul is going to be drawing a more asymmetrical tree, and Brian's going to talk about asymmetry and what happens when the daughters have different relationships. Is that correct? So Paul, we're at 11:32, so we should probably pop over to the next drawing sample.
Well, this is something, as you can just see, this is just going to keep going on forever as these things branch. And at some point, I stop measuring, and I just start doing it, drawing in these little branches freehand. And I think I had counted maybe about 8 generations in the Palo Verde trees that are around my office here. And so, should I just keep on moving, Roseann? Is it? I don't know I'm not keeping track of the time.
Yes. So I think this is like what we're doing is exploring these ideas by drawing. So what Paul's going to do next is so stop this screen share, Paul. And
sorry for going so fast.
No, this is great. This is a taste. This is, this is wonderful. I think it gave people a good look. So can you stop your...
What I want to do next is basically once you sort of have this in your hands and in your brain, I start looking at trees. I start really looking at plants and trees and becoming more and more kind of fascinated with those things that just look like chaos at first. Seeing this order within that, along with a certain amount of randomness. And now I quickly walk me through how I get back to this.
Do you want to share the photo of the Palo Verde at the same time that you are sketching it?
Yeah,
okay. So if you go to screen share, and this time just go back to basic and select your desktop.
Okay.
And then you'll open the go find your QuickTime plan.
This is another thing I really quickly, I'll put this in the chat or something. There's this wonderful poem about Hokusai. I was really glad that you mentioned Hokusai, Brian. Hokusai says, look carefully, pay attention and notice. Keep looking, stay curious. There's no end to seeing. Let me find this. This is a Palo Verde tree I just took on my phone yesterday. That's growing outside my office here. And this is an impossible thing to draw because it's just so complex. I'm going to just show really briefly. This is an example of the chaotic, stochastic, or really asymmetry that's very expressive. This tree, the trees are just very expressive. let's see, now I want to get.
So you want to click on your QuickTime player and get that going. And move that now so that it.
Okay, so you can see this.
And yeah, just move your window. There you go.
I'm actually doing this on a laptop. I would like to somehow look at this while I draw. It's gonna have to be sort of small. Just briefly, there's this book that I'll put out on the I will send this out, or I'll put it on the chat. This is sort of like, if there's a textbook for this workshop, it would be this. This guy, Rex Cole, New Trees and New Plants. And there's a quote that he has in here that I just wanted to go over really quickly. That's an artist's viewpoint of exactly the same kind of thing that Brian was talking about. He says, unity is gained in nature by a consistent adherence to a certain type of line in all the individual trees of a species. On one, the branches form lines that are set with another at a small angle. In another species, the angle is larger. Some species exhibit slight curves or full curves, or the boughs form elbows. One type of line is retained by each. Every branch takes its place in the scheme governed by the prevailing angle, curve, or straight line that distinguishes it, and a harmony exists that could not be if each bough and twig had a law unto itself. Though every twig is formed in the plan of its neighbor, and every bough is but a twig grown old, yet endless variety is obtained by our different point of view for each. So this is just some of the beautiful drawings that Rex Cole has done. He was a landscape painter, British, of course, because of the beautiful language that he used to describe this. But just take a look at this book, and you'll see an artist's view, which in a different kind of a way reflects sort of the scientific kind of view that Brian was
Hey,
talking.
Paul, can you move your cursor off that portion of your screen, and then that volume thing will go away.
Yeah, so.
Just click off of it. Yeah, move your cursor off the QuickTime. There you go. There you go.
There I go. All right. So I'm gonna just break down this tree. I don't know how much time we have.
You have about 18 minutes.
I'm just gonna quickly break down this tree. In a form like, of course, this tree is impossible. But I'm gonna make sure I can fit it. So, but say this Palo Verde trees, they have multiple trunks, and you just see the as Cole explained in this book at another place, he looked at drawing these trees in the same way that another artist would draw nude figures. The complexity and the beauty of the forms and the subtlety of all of these things are just as intriguing in plants as they are in the human figure. But just to break down a tree, I don't know if I have to do a good drawing, but I'm going to do a drawing of how you would just break down the complexity of a tree. You just want to know the vertical, and there's a canopy here that which is going to be different. It's gonna. I sort of start with a circle just to see where I am, and a large part of this canopy fits within that circle. but there'll be little bulbs here where I can see that this tree forms kind of a parallelogram here, or kind of like an odd, what would you call that, a trapezoid shape. There's little, and then there's sort of major lines, and I don't really want to look, I just want to have a suggestion of knowing where that vertical line is. So the basic formula for a tree is just kind of a line, you know, in a circle for the canopy shape. And I see that there's just basic lines that are going up. There's major lines, and each of those lines goes into, since it's continually branching, there'll be smaller and smaller little sets of branches and shapes that'll come out of these major lines. And I see that there's a couple like going down over here and down over here. And there'll be, as the branches start spreading out, you'll see that they're getting thinner and thinner into just kind of this mass. There's like a cool trunk coming up here, and another like really cool little branch that comes off like that. And the lighting. So the way I would look at trees is trees that are a function. I'm going to just talk. I really can't draw and talk at the same time, but trees are a function of time. Whenever you're looking at any kind of a plant, you've seen that plant at one point in time. In fact, even at different times of the day, the lighting will be different. But you've seen, you know, 5 years earlier, this tree would look quite different. It's because the growth of the tree equals the form. The form is just basically the growth of the tree using these same kind of patterns over time. And I'm not sure exactly how all these beautiful curves are produced, but it's just sort of the biography of this tree is told within all of these curves. Some of these, I think, like if this branch is curving down like that, at one point there was another branch branched off that just disappeared or fell off. And sort of what's left is like the visual story of the life of this tree that remains as the new growth is always going to be thinner by a certain volume from the larger trunks. The main thing you just got to keep in mind is that don't make any of the daughter branches the same thickness or any thicker than the mother branches. So there's sort of like a second ramification here, and I'm just going to really quickly show just a way of thinking about a tree when you're visually. By doing this, by drawing these trees, you understand them in a way that is profoundly different from just walking and just noticing, well, that's a tree, it's an oak tree, it's a Palo Verde tree, or identifying trees. If botanists would draw plants, they would have a deeper, they would, combined with their scientific knowledge, they would have just this deep understanding of just sort of following the lines with your hand. It's a very like, it's almost like a very emotional sort of intimacy you develop with the tree when you're spending time with it, which is the same kind of thing that artists say they develop in drawing portraits. You become just very, very knowledgeable about that person, drawing their face. And I believe that the branches in this tree kind of show the life story of this tree. All these curves will be different in every tree, and some of it will be just random. Some of it will just be the environment. So I've got, like, as these things start ramifying, I'm getting these sections of branches, and just find these might, these just might be kind of a tone, the way Leonardo drew these trees, but you don't want to draw contrary to the growth of the tree. All of this thing is growing out. It's just kind of growing out from expanding and growing from this base seed. All your little lines, even if you're just sort of quickly showing them, you're going to want to follow that growth pattern of the tree. And I'm going to somewhere, you even might even want to just shade this in, like with a thicker, as you could see in some of Leonardo's trees. At some point, there are some actual leaves on this Palo Verde because it's the time of year, spring, and there's little leaves that are tiny, you can't even see them. But there is like a little bit of there's a little bit of leaf stuff kind of covering this branch. And you might just kind of indicate that in. So branches sort of come in and out of visibility. And there's a certain all these things are all coming from the same branches. I'm not really paying attention to the angle, but if you have that angle in your mind, Palo Verdes are really consistent in that sense. Although the one next to my door by my office here has like almost 90-degree angles. It's coming out, so lots of trees will be very rebellious, and they will try to destroy your theories as best that they can.
Hey, Paul, while you're sketching in those details, it's really cool how that's capturing the gesture, the shape of that tree. It does look like a Palo Verde. Maybe Brian can talk a little bit about the biology of Palo Verdes and how this asymmetrical form grows, and maybe a little bit of the fractal element here.
Sure, I can jump in as Paul is doing his magic. I have to say I'm very impressed. I'm enjoying watching this emerge. Yeah, a few things I wanted just to talk about here as Paul is drawing. I think several of you, if you kind of take what we've been talking about in terms of branching rules, and then you go out and you start drawing kind of individual trees, one thing that you'll immediately, I think, kind of identify is that there's going to be some variability in these different rules. And indeed, that's the case. So even though we've kind of focused on, for example, like the Leonardo rule, if we start actually measuring different tree species in different environments, what you actually find is some tree species have their branching rules differ in one direction than from another species. And we start to see this kind of this variability kind of in nature in these different branching rules. And it's really interesting because the variability is actually kind of the spice of life, right? The diversity of life, and natural selection is trying to push, different species, to better fit into new environments and interactions between other plant species and their pollinators and microbial species in the soil. That kind of selects for different kind of forms and functions, and natural selection is always kind of playing with these rules a little bit to see how far away to kind of to bend them or to break them. And so there's some pliability, there's some variability in these rules once you start going out and looking. But what's interesting is that variability is highly constrained. That is, it's not a total free-for-all out there. Leonardo's rule and the volume preservation rule, that's held up pretty well. So on average, when we look across species, that tends to be the case. But if we start looking at some specific species, we start to see some deviations. And Palo Verde is a really interesting kind of case of that. So one thing that you'll notice almost immediately with Palo Verde is the name, which in Spanish literally means kind of green wood, right? So verde then meaning green. And the stem is green in color, obviously. That color reflects the fact that these stems are actually photosynthetic. Okay, so they're kind of functioning almost like a leaf, which is kind of weird. So Palo Verde plants, their stems operate differently than, say, an oak tree or a maple tree or a pine tree, right? Where you have this nice thick bark that's usually gray or orange in color, right? And then they branch all the way out then to the tips then where you have then the leaves, the green leaves. Well, Palo Verdes are a little different. They're kind of a cross between kind of like a cactus and a tree, because their stems perform photosynthesis, right? Just so like a cactus. And in fact, a cactus plant, one thing you'll notice with cactus plants, they don't have any leaves. Well, actually, that's not true, because their leaves have been so reduced in form, the spines on the cactus, that is the same thing that's analogous then to a leaf. So the leaves then on cacti have been so reduced kind of over time, they then basically perform a function of protection, which is the spine. So on cactus plants, spines then are leaves, which is interesting. Now, a Palo Verde plant is kind of on the evolutionary kind of trajectory of reducing the total leaf area in these very arid environments. And so Palo Verdes put on leaves, these very small little leaves, only when it's wet outside, and then they lose them. But then the rest of the year, they kind of act like a cactus in that their stems are the main organ performing photosynthesis, okay, which is different than other trees. And because of which, a tree then where the stem is no longer providing just the function of kind of biomechanics, that is making sure that it holds up the rest of the tree, as well as just supplying water and nutrients then to the leaves, there's another function that the stem is providing, and that is of doing photosynthesis. And that actually, it turns out, kind of alters these rules a little bit. And so they don't follow the Leonardo and the volume preservation rule as strictly as other trees do. And because of which, if you notice, the Palo Verde, the links are more extended. And so the length rule, as you go from the base of the tree out to the tips, the links on the Palo Verde are actually more extended out. So it's like you take that nice symmetrical tree that Paul was drawing earlier, and that rule of keeping, the 0.7, the proportion as you go, the lengths, that rule is actually extended out a little bit, so that as you go from mother branch to daughter branches, if you notice on the Palo Verdes, it's more elongated. We call those leggy trees. That is, for the given size of the branch, it's this extended length then to them. And so it kind of gives it this weepy, kind of extended kind of look to it. And part of that has to do with the Palo Verde branching kind of rapidly gets to these very fine branches very quickly. And so a lot of the surface area, if you look at that Palo Verde picture on the left-hand side there, if you kind of notice, it's kind of hard to see kind of the bigger branches in there, because a lot of the branching kind of surface area is in those extended kind of smaller branches at the end, right? And so if you try to put your hand into a Palo Verde, it'll get scratched up pretty quickly, because there's all these fine little branches kind of in there. That's because it has really leggy branches. So very quickly, as you go from the base to the tips, the Palo Verde kind of quickly dissipates into this fine mist of these fine little branches. And all those fine little branches increases the surface area of the stems in order to provide photosynthesis, because Palo Verde, the actual leaf area is very, very small. So that's an interesting little modification in different environments, that natural selection, depending on the different functions then of the branches, can kind of alter these rules. So one thing that you'll notice in drawing it is like, whoa, it's actually it does look a little different from that perfectly symmetrical rule of area preserving and volume preserving. It's a more kind of attenuated, kind of leggy kind of branching then to it. So its rule is a little different. And that's because natural selection is kind of selected more for this increased surface area of these smaller branches at the end, because the branches are performing photosynthesis, not the leaves.
Oh, that's awesome, Brian. Hey, we've got a few questions. We are 4 minutes from the end. And so I'd like to get a couple of these questions out because there's some really good ones. And so, this is for Brian. Bert asks, I can imagine a structure that doesn't give a straight line on the log-log plot when box counting. Maybe that couldn't exist, but if it could, would it not be as good at transporting or catching light as a structure that is strictly fractal?
Sorry, I muted myself. So if you do the box counting exercise, one thing that's actually surprising is that a lot of structures in nature do kind of have that straight line behavior to them. But occasionally you do see objects, structures, organisms that are not described by a very nice straight line. Sometimes you get a line and then a break. And so you'll see kind of like a bend or something in the structure. And that's important if you see that, because that tells us that there's a scale break. That is, that as you go from one set of scales up to the scale break, the system is obeying one set of rules, but then once you get to this other kind of increased scale, there's another set of rules then that kick in. And sometimes we see that. There are some physical processes that only operate on certain spatial and temporal scales. But then once you get to larger spatial or temporal scales, another set of physical rules kind of kick in, right? Or another process. And you can see that then in those graphs. Also, sometimes these rules change as a function of the scale. And if that happens, then you'll see curve linearities then in those plots. And that tells you then that the rules underlying the creation of that structure are actually changing as a function of the spatial scale. And so that's kind of handy, because one thing that you want to know if you're studying a system is that, are those rules then that are creating the pattern, is it always the same? And if it's always the same, that makes the problem easier, right? Because all you need to know is what those rules are. But if those rules then are changing, so for example, as Paul is drawing his tree, he was keeping that proportion the same, 0.7, right? Sometimes we find in certain situations that actually that proportion 0.7, that changes. So, and that makes things a little complicated. And what that says is that the rule is actually scale dependent. And that makes for, there's different kind of terminologies. Sometimes people call it a multifractal or kind of, self-affine kind of structure. It just means that the fractalness is a little bit more complicated to it. But that's a good question.
Yeah, thank you. And
let me just say something.
We're running out of time here, but there is one other question, and we can stay a little bit later for those who have the time, but this question I think is good because it has some conservation and ecological value. And that was, someone asked if you can determine the fractal number or dimension for different plant species from satellite images.
Actually, good question. Well, it kind of depends on the satellite imagery. You could, well, on any image, you can calculate a fractal dimension then of the image. Okay. Now, the problem with some images is that you're not seeing the entire structure, right? So for example, if you just take a picture of a tree like we have right here, that's pretty good. You're getting a lot of the branching structure, but you're not actually seeing all of the branches. And so to do this right, you want to try to image three-dimensionally the entire thing, right? And now we have these scanners, they're called LiDAR scanners, these laser scanners, where you can kind of basically circle the tree and shoot lasers at it and look at the returns, and then you can reconstruct, the 3D structure from it. And so from a highly accurate laser scan, that's in 3 dimensions, yes, you can extract out kind of fractal dimensions then of those structures. If it's just a 2D picture, especially if it's farther away, you're going to have less information about it. But you still can, on the image, you can still generate the fractal dimension of that image, but it's not necessarily the full structure that you're looking at, if that makes sense.
Yes, thank you very much. Well, we have reached noon. Actually, it's afternoon. Paul's got some other images we will put on our website. Actually, we will post his drawing here. We'll post the image of the Palo Verde tree. Paul, if you can stop your screen share, I want to bring you and Brian back on just to say goodbye.
Mm-hmm.
Yeah, I could talk about Leonardo's drawings for a while, but
let's yeah, there we go. Let me pin Brian again, and there we go. And great. So I know we could keep going forever and ever. This is like completely amazing. And there were some good questions. We can't really keep going too much farther because we did promise people a certain time frame. Maybe just like for 1 minute, Brian, can you talk about crown shyness and that overlap, disinclination to overlap amongst trees in a forest, and how does the fractal factors work in there?
Yeah, so it's a really good question. Actually, I kind of teach an entire course on all of this stuff, so we're just kind of skimming
it down into 30
we're skimming the surface here. So so what's really fascinating is that so we just focused on individual trees and individual branching and these principles of space filling and kind of the Leonardo rule of area preserving, right? So again, natural selection appears to have acted on biological networks. So they try to fill a volume, some sort of volume, where they're extracting resources like light or carbon dioxide or oxygen, right, from a given space, okay, a three-dimensional space, and then kind of transporting that kind of through the body, right, and doing that in a way that saves time, minimizes time, and minimizes the cost of transport. And so these trees seem to be kind of great, networks, for doing that. And if I were just to kind of diverge a little bit, a lot of these principles actually say a lot about, the design of our cities and infrastructure, too, because we're horribly not optimal in terms of our own distribution networks, in terms of how we design road networks and transportation networks, like in general. So there's some very interesting kind of like parallels, to that. But in terms of things like kind of crown shyness, so the question was, how does this relate to if you want to study the canopies of forests, or how plants kind of fill in then the landscape, right? So we just studied one tree, but what about all the different trees, all the different plants then kind of on the landscape? So if you actually kind of look at their canopies, either their rooting volumes or their above ground canopies, you can really see this in forests. Trees tend to only occupy kind of separate spaces. They very rarely kind of overlap their canopies. Sometimes they do overlap, and if they do, it's often not the best for both species. They kind of get in the way, and growth kind of slows down. So in general, each kind of species kind of finds its own kind of space, and so so the canopies kind of fill in their own unique space, and the overlap between them is kind of minimal. And so there's this phenomenon called crown shyness, where if you look at the overlapping kind of tree networks of two different trees coming together, they'll kind of keep a kind of a set distance, right? It's like everybody has their little territories, right? And the branches try not to kind of like grow into each other too much. And there's so that makes sense from an evolutionary perspective, because if you have your own space, that's the space for which you kind of forage light and CO2 from, and you're not kind of overlapping in somebody else's space where resources then would be in competition with each other, right? And crown shyness then is kind of avoided in a few different ways. And so trees, as they grow, they sense the light environment. They actually can see. They sense, for example, ratios of near and far infrared light. So as you actually move closer to another object, maybe that's shaded or you get reflected light off of, trees can actually sense that, and they will actually grow away from objects that are kind of close that may be shading them. And because of which they'll try to then avoid each other. But then also another mechanism that's kind of enforcing kind of crown shyness so that these crowns kind of fill in their own space is that as the wind blows, these the branches are kind of moving kind of in the canopy. And if you've been in a forest or on a really heavy windy day, you'll see the branches kind of swaying. And what happens is that these branches will kind of hit each other, and then they'll basically, you can think of a windy day as basically like a medieval kind of Game of Thrones kind of like competition kind of out there with these branches whipping each other. And it's a real fierce kind of fight. And then those branches that are the most biomechanically more stable will actually be the ones then to whack off then the branches then of others, right? And it's in those really windy days, where it's really this Game of Thrones war of branches out there. And then when you go through on a nice day, be like, oh, look at that nice pattern. The trees, they're filling in their own shape. Well, that's actually the outcome of some pretty brutal warfare in terms of branches against branches out there. And so that kind of shapes where then these trees then grow. And so the end result is that the forest actually follows these same kind of fractal rules of filling in all the space, right? But they're all kind of obeying the Leonardo rule. And so we're then able to take these same rules of space filling and area preserving, and that's how we can then scale up. We multiply through by all the individuals in the forest to say something about, how the entire forest, suck up carbon dioxide, how much biomass is in there, what does that mean for, the height structure, for other organisms and things like that. So these rules are not just nice little rules, for drawing. We actually use them to make calculations with the flux of matter and energy and carbon and nitrogen and so on. So they're very handy. They make calculations easy.
Yeah. This is so fascinating. We could have an entire semester class, which Brian does. So I want to thank you both so much. I hate to stop. It's hard, but I think folks have places they need to be. And we have recorded this. I will be sending you the link to a private webpage with all the resources. So all the images, links, books will be on there for everyone. Links to Brian's website and Paul's information. And anything mentioned in the chat that people are sharing right now, I'll also put on there. So thank you both so much. This was just mind-blowing. And next week we have, or next month, we have river meanders, and we will be looking at that through the lens of river restoration and the Santa Cruz River in Tucson. And how
similar rules in river networks too, by the way.
Exactly. So we're going to look
physics are a little different, but it's
fluid dynamics. And but as a river is reborn, and watching this little baby new river come back, forming its meanders, and it'll be really, really fun. So quite different. So everyone, I hope you enjoy the rest of your weekend. Brian and
thanks
Paul.
Everyone. I want to say thanks to Paul. Paul, that was awesome. Based on very little bits of communication here and there, I'm very impressed with what you've been able to do. So yeah, it'd be great at some point, to hang out for a little bit, hopefully up at Tumamoc.
Yeah, with a glass of mezcal.
Even better. Right. Okay.
Awesome. Okay, guys.
Take care.
Thank you very much, everyone. Bye.
All right. Bye-bye.
Bye.